Okay, good afternoon or good evening, everyone. It's nice to have you here. Here is a moss that every beginner and every other person should get to know because it's very, very common. Many of you probably already know it, but we're going to introduce it here. It's called Roaldia revolutum. It used to be called Hypnum revolutum. Roaldia reflects more accurately its relationships to other similar mosses. And Hypnum revolutum you can just use as a common name. Everybody knows it by that name. If we go into any of our dry New Mexico forests and close your eyes, bend down, and grab something, it's very likely to be this. And it looks very much like this, kind of a rusty, golden, brassy looking type of moss. You can see that it's a pleurocarp. So here is the main stem. Here are the dense lateral branches coming off. And so most of what you see, all of these are branches here connected to a main stem hidden in there somewhere. So these are the branches of the pleurocarp, and it forms these kind of low mats or mounds, and it's spreading. It's often referred to as having a braided look to it, and I guess you can imagine we only had sons in our family, and so we didn't braid pigtails or anything like that, but I guess you can imagine this braided look here. And all the leaves are downturned. You can see how they're all kind of curved downward. And you don't really see the tips of the leaves except maybe on the edges. So anything that looks like this, and that color in some conditions is very characteristic also, but Roaldia would be a good thing to think of, at least at first shot. Let me see if I can advance. Seems to happen every time. There we go. But it is often green as well. So here are some more greenish shoots. Here in the upper left, we can see the downturn, plaited or braided look to these shoots. Each one of these leaves, if you turn the little branch over, here are the leaf tips. So these two, we're looking, I've turned the leaves over and you can see the leaf tips curling up. So they're strongly falcate, very much overlapping. You don't see the stem. The stem leaves would be right here. They're very similar, except a little larger. Here you can see the downturned leaves. So between these two pictures, that says it pretty well, but you'd better look at some other things also. Here are the leaves that are falcate, curved to one side. You can see that they're often plicate or folded. So here is a fold right here. You can see a fold right there. You don't really see it so clearly in this picture, but the edges are rolled over. But one thing that does stick out right here are the toothed margins. And that gets to be a little bit of a problem. Let's look at the next one. Well, I'll just keep going with the slides. You look down at the costa at the base of the leaf, and here are two leaves. And so you have a double costa. So this is right down at the base, and there's one branch, and here's the other one, the other branch. This is about as long as it gets. So we're only seeing the bottom quarter, maybe, of the leaf. And it can be quite a bit shorter and sometimes even nearly absent, and you have a hard time seeing it. It's quite common to see one costa branch long and one costa branch shorter like that. So these plaited leaves, downturned, kind of a brownish reddish color, and then you get it under the scope and you see this double costa. You're in this group of old Hypnum things. And then as you start looking closer, it's called revolutum because the leaves are revolute. That costa is hard to find here. And this is done more, this cross section is down toward the bottom of the leaf. but it's very difficult to pick out. Here's the other edge, revolute. And they're often toothed. Now, this creates a little bit of a problem because the literature commonly says that the leaves are entire or that the apex of the leaf is entire. But as I got looking around, it was hard for me to find an entire leaf. What you find easily is the leaf rolled over, and if you don't look carefully, you might think that's the edge of the leaf. But as soon as you look more carefully, here's the actual margin, and the teeth are quite prominent. Here's a plication right here. So I got interested in this tooth versus entire. So here's an apex, and you have this very prominent toothing. That's a little bit contrary to how we find this scene described. And sorry to say, even in our book, we describe it as that, based on other people's writings. The cells themselves are kind of long, narrow, a little bit worm-like, I suppose. As you focus up and down, it's very common to find these tips of the cells that are rising up. So they have a little projecting end, and you can see that. This is just a single, this is not a stacked photograph, so a single focal plane right here, and you can see those little bumps at the end. They almost like, well, it's like the edge of the leaf is turned up and almost like a little papilla. They call that prorate or various other things, but that's kind of what it looks like. Now, this idea of whether the margins are entire, especially at the apex, I started looking at a bunch of them. I had a hard time finding an entire leaf. Here's one. When it was particularly drawn out, it was pretty much entire, but most of what I found was this sort of a thing here. So we need a little bit of an adjustment is how we think of these, at least in my experience. And there you have very prominent teeth at the apex. You go down to the base, and in the corners of the leaves in the alar region, these little squarish alar cells in a rather small cluster, especially when we compare it to other things. Here it looks like maybe one of the costa branches is here and one of the other one is right there. And they're homogeneous. That is, we don't have two kinds of alar cells. Here you can see the margin rolled over all the way down to the base, and there is the edge going right up there. Here we can see those long worm-like cells. They shorten up a little bit down here. Here are the alar cells down here. Well, as you might expect, there are things that look a little bit like this. Here are the ones in particular you might want to think of. Of course, our plant that we're concerned with, which you'll see a lot of. So it's not a bad first choice, is to think, well, this might be the old Hypnum revolutum. All of these have been placed in the genus Hypnum. Now, phylogenetic analyses have pulled out these three into separate genera. One thing that will help is the presence or absence of a capsule. Here's our typical Roaldia revoluta, mat forming. You can see the branches. It's not a close-up, but that's a pretty good look. If we see a bunch of capsules, then you want to think twice, because capsules are fairly rare in Roaldia. That's because it's dioicous, and so it produces the sexes on different plants. And Joachenia produces them both on the same in proximity, and so capsules are easily produced. The branches themselves look kind of similar. So you check out the capsules. And then you'll want to look at something called pseudoparaphilia. This is a little bit advanced. You have to do some dissections. It takes a little practice. These are leaf-like, microscopic leaf-like structures associated with the buds or the branches of the stem. So they are false paraphilia, and paraphilia are little hair-like or branching structures that go up and down the stem. Pseudoparaphilia are always associated with the branch, kind of analogous to bud scales on vascular plants. So here is the little bud area that a branch will emerge from, and this is the pseudoparaphylium, and a cluster of these long uniseriate pseudoparaphilia. So this is the single thing in New Mexico left in the genus Hypnum. All the others with these broadened foliose, leaf-like, here's a very broad one, pseudoparaphilia, are placed in other genera. This is actually quite helpful. Things can mimic other things quite easily. At first glance, it looks like Roaldia. 80% of the time, you're right on. But then when you start to have second thoughts, check out the pseudoparaphilia. It requires a little bit of dissection. You grab a stem that has some branches coming off, carefully pull off the branch, and then individually pull off the leaves where that surrounding that branch or up and down the stem. So there were leaves associated with these areas, and we've pulled them off, and that's how you see the pseudoparaphilia. So that's helpful. Kind of an advanced feature, but something good to have on our minds.
Kelly,
Yes,
can I have a question about that?
sure.
If we could go back. So Buckia and Roaldia, how do you differentiate them? I mean, yes, those two look somewhat different because the one on Buckia is larger, but in an overall sense, I mean, they're both kind of multicellular. They're both kind of lanceolate shaped. How are they distinguished shape-wise?
Buckia will not have the revolute leaves, usually.
Okay.
And
Okay.
also the alar region.
Oh yeah, I
It's
was
massy
asking
in Buckia.
in
And
particular.
here's Roaldia.
Right. I was just asking particularly, would you say that those pseudoparaphilia are similar or are they significantly different?
There's enough variation that they're not always this distinct.
Okay,
But in
that's what I was
general,
really getting at. Okay.
Buckia tends to be a little more foliose, and
Mm-hmm.
this is a little more like, but it's close enough, as you can see
Okay.
that you may not be able to tell, but there are just regular leaf features that will help you with Buckia.
Okay. Yeah. Gotcha.
This is the difference here between Hypnum and these others,
Yeah.
because Hypnum cupressiforme has something like 7 or 9 varieties, lots of variation, but the pseudoparaphilia hold true pretty well. So this can be very helpful.
Is it weird that the chlorophyll is towards the cellular junctions there? Or is that just I don't look at mosses usually, but it's just a product random, or is that like a thing?
Like what? I'm not
Like
sure
in
what
your
you mean.
Hypnum cupressiforme picture,
Uh-huh.
chlorophyll is clustered towards the junction of the cells,
Down. Oh, right in here
like a thing, or
and right in here.
yeah.
I'm not sure what causes that. That could be something to do with drying.
Okay, yeah.
And so as the cells dry, they tend to kind of desiccate a little bit. I'm not sure totally. Okay, should we move on? I just have a few more slides. So we look at the alar cells. And so as we've already noticed, here is Roaldia with a homogeneous, comparatively small cluster of quadrate cells. Buckia, which has actually different leaves, and I didn't picture them because we're just looking at Roaldia, but the alar region is quite a bit more extensive than in Roaldia. Here's the one that causes problems with both of these, but the alar regions are quite different. You have greener, smaller cells up here and larger hyaline cells down here. And that's actually a pretty good feature, but you'd better look at more than one leaf. So pull off carefully several leaves when you're looking at alar regions and make sure you're getting the base of the leaves. So alar cells help us in some of these problems. So here's a quick summary. I think this will be online, and if you want to go back and kind of look at some of these, that just sort of reminds us of what we've gone over. Your
Is this
first
going to be in?
glance is going to be a good one, but sometimes you do have to get a little bit more detail. Yes, I heard. Was there a question?
Yeah, is this going to be on the test?
Well, I've got nearly 40 years of college teaching, so I don't answer those questions.
Yay.
Okay, that's the end of my little presentation. If you haven't seen Roaldia, common name Hypnum revolutum, you will, and hopefully you'll recognize it. It's a good one to get to know. It's throughout the western United States in all the forests, and then it goes up in elevation and sneaks down into the kind of the drier but shaded habitats. And here's a lot of different ways to say good luck, beginning with English and ending with Hawaiian.
Hey, Kelly, could you go back to the very first picture, that beautiful brassy thing? Yeah, that. Um, so it has, I'm just trying to figure out in my own mind. So it has the beautiful yellow golden endings to each branch, and those look a little different. Are those just new leaves that are
These are
different?
newer leaves.
Okay,
I'm guessing that this color is environment induced,
okay,
much like changing of leaves, in the fall, that kind of thing. Because we do see them frequently, like this.
okay,
And so when you go into the drier New Mexico mountains, you find these, we find most of our mosses in the dry state. And so this is how we often see them. And these are the younger leaves, undoubtedly. And I guess I don't know exactly the duration of this color and all the parameters of that. But we
wow,
do
this,
find this sort of thing frequently. And there are some other mosses in other families that will look just like this.
yeah,
They may not have this arrangement of these very regular pinnate, but this strongly falcate curving leaf, Sanionia, can mimic this. So once you get it under the scope, you look for the almost here, it's almost an absence. There is a costa, but they can be very faint. Anyway, other questions?
I have one more question, Kelly.
Sure.
When you read about paraphilia, which occur along the stem, and pseudoparaphilia, which occur at the buds or the branch bases, I never read anything about them being on the same plant. Do some plants have both?
I'm thinking here. I guess I don't know for sure that the answer to that, but just off the top of my head, it's kind of like what you have thought of, that we either have paraphilia or pseudoparaphilia, but I'll try to look that up a little bit more.
Thank you.
I want to go back to the last slide if we can. The beautiful golden green. Your first slide.
First slide.
Yeah, that what struck me first about this is that that's totally opposite from vascular plants, at least the ones that I've studied in my career, wherein most flowering plants you get red, very, very young leaves, right? So towards the tips of the growing axis. In the neotropical forest, you would get red, very, very immature leaves, and you would have green leaves towards the base and internal portions of the plant. That's like totally the opposite. Although I guess in this case, the red is still kind of sheltering the green, but right. There's been studies of this in mosses. I was involved in one of them with John Shaw. I've worked on anthocyanins in plants, and we kind of tried to dive into it in the world of mosses with their group. And what we found was that the anthocyanin, the red pigment producing pathway, doesn't exist in mosses. In fact, what we can find is that it's first intact in seed plants, which is
Okay.
cool.
Great.
But that these red producing pigments in mosses are still part of the flavonoid pathway and presumably serve the same function that we talked about, right? These protective kind of things. So anyway, I just wanted to point out, it's just when I very first saw the slide, I thought it would be opposite, that the red leaves would be towards the tip, the growing axis, and that the basal leaves would be the older green, more protected things. And this, I don't know, maybe just as a product of, in vascular plants, you get these large, hardened, long-lived structures that can accumulate all kinds of products and whatnot, versus in bryophytes, these are very thin. I don't know, maybe temporally seasonal. I don't know. I'm not really sure, but it just struck me as totally opposite of how I would have painted that picture in my head from what I know about vascular plants.
Yes, thank you for adding all of that. I really don't know very much about, that phenomenon in mosses. So
is this normal? Like when you guys go out in the field, if you see red and green, is red kind of older and green is newer growth, or not?
In mosses?
Yeah.
I'm thinking of Platygerrium that has this same kind of pattern. And I'm just trying to pull things up, from memory. And I associate this so much with the, with Hypnum revolutum. I guess I don't know. I can't address that.
Yeah,
some others have some ideas.
I think in liverworts, generally the growing tips are bright or the brightest green, and then secondary pigments, but are towards the base of the stem, the older parts are darker. Now, I don't know if it's because of the pigments being hidden by another pigment, which is probably the case in some cases, or if it's just that there isn't much chlorophyll there anymore. But definitely I associate growing tips with bright green and darker pigment farther back.
I should know this because I've been in enough sphagnum bogs, but in peat bogs, is it the same? Like what we're seeing in Barbara, what you just described? Is it red is older and green is younger, or is it the opposite?
It's a little different, right? I mean,
that's certainly true for Scorpidium.
Yeah.
Has the red, basal parts of the plant,
but they often talk, don't they, that in sphagnum bogs, if there's a difference in color, those that have red and then red and green, the green ones will be in shaded areas and the red ones will be bright light. So it's that photo ultraviolet or wave sun protection kind of thing. Centrichia is that way too. I mean, in the shade, you can see really just plain green centrichia, and then other times it has a lot more red in it. So there's that. More food for thought.
The more I think about it, more liverworts that are reddish in color, and there are some, there's quite a few, in fact, Frulanias and things that grow, and the more exposed, the redder they'll be. Redder or browner, for sure.
There were some more questions there that you posted, Erin, that look pretty interesting. Sharp tips for rain, water shedding. Are these taxa, the falcate-leaved ones, overrepresentative in wet environments? That's a good question. All these ecological questions. I don't know that one.
This plant grows in dry environments. So generalities about the leaf shape probably, have some merit, but I don't know about this particular species.
Right. Then
what
there's
about that family, or is it Hypnaceae? I'm making that up. What about that group as a whole?
These all used to be in Hypnaceae, and now this one is pulled out in the Pileasiaceae, a little sister group. So basically related Hypnaceae.
And are they
very widespread, lots of different environments.
Okay. All right.
Yeah, there's Japanicladius. That's two different species. One's falcate and one isn't, but they have similar wet environments. So, ta-da.
Yeah, good point.
Okay. Thank you, everyone. Thank you for your comments.
Thank you. Thank you, Kelly.